Plague DNA inside ordinary teeth exposed a disaster the chronicles barely knew how to name. The graves were not royal tombs. They were not battlefields under banners and armor. At East Smithfield in London, between about 1348 and 1350, rows of ordinary bodies were placed into emergency ground while the Black Death moved through the city. Most of those people left no individual name in the record. A clerk might count the dead. A chronicler might write about fear, carts, bells, empty streets, and parishes overwhelmed by burial. But the person in the row, the child beside a parent, the laborer wrapped in haste, the migrant who died far from home, often disappeared into a number. Then scientists looked inside their teeth. A tooth seems too small for a catastrophe that killed across continents. Yet dental pulp, sealed inside the hard shell, carries traces of the blood vessels that once passed through the body. If a person was infected before death, fragments of the microbe could be trapped there, protected for centuries inside the jaw. That changed the grave. The burial told archaeologists who had died together. The tooth told them what had been moving through the victims while they were still alive. The written record could describe a plague year. Ancient DNA could point to the biological killer inside one particular human body. The first model broke there. For a long time, plague graves were treated mostly as historical scenes. They belonged to chronicles, tax records, churchyards, and memories of sudden death. Ancient DNA made them colder and more intimate. Biological cold cases where the dead themselves could still bear witness. The caveat matters here. Pathogen DNA is not folklore. It is not a curse dressed up as science. It is not a symbolic label for every medieval disease that terrified people. In this case, it means recoverable genetic material from microbes that infected living bodies before those bodies were buried. The bacterium at the center of the story is Yersinia pestis. It is the organism behind plague, including the medieval Black Death, and earlier pandemics that struck long before London dug its emergency cemetery. Under a microscope, it is not dramatic. In history, it moved like a shadow through homes, ports, roads, and crowded rooms. Dental pulp matters because infection is not only something on the skin or in the air. Blood moves through the body. During severe infection, bacterial DNA can end up in the blood and remain locked inside tooth roots after death. Centuries later, a laboratory can sample that inner chamber, separate the human DNA from microbial fragments, and ask a blunt question. What killed this person? That question has to be handled carefully. Ancient plague DNA does not prove that victims were weak, guilty, dirty, or somehow destined to die. It does not make medieval people lesser than modern people. It does not turn hunger, poverty, crowding, war, trade, or fear into moral failure. It shows contact. It shows a microbe moving through human networks. It shows that crowded societies, grain stores, animals, fleas, ships, soldiers, markets, and roads could create pathways no person fully understood. A bacterium does not need a kingdom to confess. It only needs a root. That is why the evidence is disturbing. It does not blame the dead. It reveals how easily ordinary life can become a delivery system for something invisible. In a plague cemetery, the skull is not only a skull. The jaw is not only bone. A tooth can be a sealed message from the bloodstream of someone whose voice was never written down. Once the method became clear, the next shock followed. Some graves that history had treated as broad catastrophe could now be tested at the level of individual bodies. And some plague stories that seemed centered on emperors, capitals, and famous cities suddenly spread outward into places the chronicles barely held in view. East Smithfield became one of the clearest proof objects. London created that burial ground during the Black Death because ordinary churchyards could not take in the dead fast enough. The cemetery was planned under emergency pressure, with trenches in rows instead of the slow, named rhythm of normal burial. Archaeologists could see order, but also speed. The ground looked like a city trying to remain human while numbers outran ritual. In 2011, researchers working with remains from East Smithfield reconstructed a genome of Yersinia pestis from medieval victims. The study appeared in Nature and became a landmark because it did more than suggest plague. It rebuilt the genetic fingerprint of the bacterium from the bodies of people buried during the catastrophe. The process was careful and destructive in only a tiny way. A tooth was taken from a jaw. The hard surface was cleaned. Powder was drilled from the protected interior. That powder held a mixture of human DNA, environmental contamination, and broken microbial fragments. The task was to find the ancient signal inside the noise. Imagine a lab screen filled with pieces of text from a ruined book. Most fragments belong to the person. Some belong to soil. Some belong to microbes that arrived after burial. But a small set of fragments begins matching Yersinia pestis. Line by line, the pathogen's genome comes back into view. That is where the grave changed from a tragic historical site into a biological crime scene. Not because a person had committed the crime. The killer was a bacterium. But the evidence had moved from rumor and description into the body itself. London's dead no longer stood only inside a story told by survivors. They carried the pathogen signature in their teeth. The second layer was stranger. The same kind of evidence reached backward to the Justinianic plague, the pandemic that began in the 6th century and struck the Mediterranean world during the age of the Eastern Roman Empire. Written memory often pulls that pandemic toward Constantinople, imperial politics, armies, taxes, and court life. That is the gravity of elite history. It remembers palaces first. But ancient DNA has confirmed Yersinia pestis in 6th century burials far from that imperial center, including graves in Bavaria. Those skeletons did not need a court historian standing beside them. Their teeth carried the proof. That matters because it changes the map. The first pandemic was not only a story of one capital, one emperor, or one Mediterranean catastrophe. It reached into communities that written sources could easily flatten or ignore. Villages, cemeteries, and regional populations became part of the same biological event. A grave in Bavaria does not look like Constantinople. It does not carry the same political drama. Yet the bacterium connects them. It shows how a pandemic can be preserved unevenly in writing, but more widely in bone. This is one reason plague graves are so unsettling. The written record often narrows attention toward people who had scribes around them. DNA keeps finding evidence in people who had no scribe at all. By the time researchers compared medieval and earlier pathogen genomes, a larger pattern appeared. The DNA did more than identify the killer. It began to trace the roots that carried it. The old plague story was too small. For many people, plague begins in the imagination with medieval Europe. Black clothing, church bells, carts of bodies, and cities shutting their gates too late. That picture is powerful because the Black Death was real and devastating. But ancient DNA has pushed the story much deeper than the 14th century. Researchers have found ancient Yersinia pestis DNA in remains from the Bronze Age and even earlier periods across Eurasia. The evidence appears in regions connected to steppe-associated populations, in northern and eastern Europe, around the Baltic, in Scandinavia, and across parts of central Eurasia. These graves belong to worlds without the famous plague narratives that later chroniclers left behind. That is where the old map failed. Plague was moving before the Black Death had a name. It was moving before medieval physicians argued about poisoned air and divine punishment. It was moving before many of the societies it affected had written records able to describe what was happening. The DNA forces a different scene. Instead of a medieval city, picture a prehistoric settlement near a contact zone. Herds move across open land. Families carry tools, grain, hides, and memory. A burial is dug for someone who died young or suddenly. No chronicle records the fever. No parish list preserves the name. Thousands of years later, a tooth still holds fragments of the bacterium. Those fragments do not prove a whole civilization fell from plague alone. The evidence points in a narrower direction. They show that Yersinia pestis was present in human bodies at times when populations were moving, mixing, fighting, trading, and reorganizing across large distances. Migration is not a simple line on a map. It is people, animals, belongings, food stores, parasites, and pathogens traveling through the same world. Moving herders could carry more than ancestry. Trade paths could carry more than amber, metal, grain, or cloth. Settlement networks could move language, tools, and disease through the same contact points. The disturbing part is not that ancient people moved. Movement is part of human history. The disturbing part is that the networks that make societies larger can also give a microbe more doors. A crowded settlement is not automatically doomed. A trade route is not automatically dangerous. A migration is not a plague engine by itself. But when hunger, violence, climate stress, displacement, animals, fleas, and dense living conditions overlap, a pathogen can travel through the cracks. That is why ancient plague DNA changes the way collapse is studied. Archaeologists already look for burned layers, abandoned houses, weapon injuries, changes in diet, climate pressure, and population replacement. Ancient DNA adds another witness. It can show that at the same time people were moving and societies were under strain, a deadly bacterium was also present. The genome kept one more record. Some collapses may have begun before anyone had writing to describe them. Some villages may have emptied without leaving a sentence. Some families may have vanished from the archaeological record as a change in burial pattern, a broken settlement sequence, or a missing generation. The graves do not give a simple answer. They leave a harder question. What if part of early social collapse was biological, but not in the dramatic way later stories imagined? What if disease moved quietly through contact zones, adding pressure where communities were already strained by conflict, hunger, or migration? What if the first witnesses were not historians, but teeth? A single prehistoric grave can hold that question more powerfully than a palace archive. Picture the scene without the comfort of a famous name. A body lies in a grave from a world before newspapers, before death certificates, before doctors could name bacteria. The person may have belonged to a farming community, a mobile herding group, a settlement at the edge of changing populations, or a family caught in a period of movement. The grave contains a skeleton, perhaps a few objects, perhaps nothing that tells us what the person feared in the final days. The jaw survives. In a modern lab, that jaw is placed on a tray under clean light. The tooth is small, almost disappointing at first. It does not look like a monument. It does not look like the key to a pandemic. It looks like something any person could lose. Then, the tooth is drilled. A tiny amount of powder comes out of the root. Inside that powder are broken molecules from the human body and from the world that entered it before death. The sequencing machine does not see a tragedy. It reads fragments. It compares patterns. It tests whether the damaged pieces match a known organism. On a screen, the fragments begin to align. Yersinia pestis. That moment is disturbing because it is so quiet. There is no dramatic confession, no royal decree, no battlefield trumpet. A person who left no written name suddenly becomes part of plague history because a bacterium left evidence in their tooth. The second layer was stranger. Elite history preserves kings, battles, monuments, and treaties because elites had the tools to record themselves. Their stone survives. Their coinage survives. Their victories are carved, copied, translated, and argued over. Ordinary victims survive differently. They survive as skeletons in rows, as enamel, as collagen, as isotopes, as microbial DNA hidden in the protected spaces of the body. An infected child does not need to be named in a chronicle to have lived. A migrant who died in a strange town does not need a parish clerk to become evidence. A servant buried under emergency pressure does not need a family document for the genome to remember that something passed through them. That is the moral unease in plague DNA. It gives back information, but not comfort. It can identify a pathogen, but not the last words of the person who carried it. It can show a route, but not the face of the family waiting nearby. It can prove infection, but not the fear inside the room where fever began. Still, the evidence matters because it corrects the shape of the record. Plague history is often told from above. Emperors terrified of empty tax rolls, cities terrified of trade collapse, churches overwhelmed by burial, states terrified of disorder. Ancient DNA tells the story from below, through the bodies that made the numbers real. East Smithfield does this at a medieval scale. Earlier Eurasian graves do it at a prehistoric scale. 6th century burials in places like Bavaria do it at the edge of imperial memory. Each case says the same thing in a different century. The dead were not background, they were the event. The tooth does not give them biography. It cannot restore their language, their work, their quarrels, their hopes, or the exact path by which infection reached them. But it can testify that the catastrophe entered their blood. It can place them inside the history of a microbe that moved across human worlds long before modern medicine could see it. That is why plague graves are not only about plague. They are about how history chooses its witnesses. A monument speaks loudly because power made it loud. A tooth speaks softly because biology preserved it by accident. Yet sometimes the tooth is more honest about who suffered. The human scene opens the deeper question. Why did the same bacterium keep appearing across centuries and continents? The answer is not that plague stayed unchanged, waiting in the dark for humans to notice. Ancient genomes show that Yersinia pestis changed over time. It had lineages, branches, losses, and gains. Like human populations, pathogens also carry ancestry. Their genomes preserve their own movement through time. This is where the evidence becomes even more disturbing. The medieval Black Death was caused by a form of Yersinia pestis that could move efficiently through the conditions of its world. Fleas, rats, grain stores, crowded housing, ports, and long-distance trade all mattered in the later plague system. But earlier ancient plague genomes do not always look exactly like the medieval form. One gene often discussed in plague evolution is YMT. That gene is connected to survival inside the flea gut, a key part of efficient flea-borne transmission in later plague. Some ancient Yersinia pestis lineages appear to lack features associated with the fully developed flea-based cycle seen in later periods. Scientists are still refining the model, and the details matter. Earlier plague may not have spread in precisely the same way as the Black Death. The caveat matters here. Finding Yersinia pestis in an ancient tooth does not automatically mean a medieval-style outbreak with rats, fleas, and citywide mortality. The bacterium's behavior could vary depending on its genome, its host, its environment, and the human world around it. The same name can hide evolutionary differences. Yet that makes the graves more troubling, not less. They preserve a record of a pathogen becoming adapted to human networks. Across time, Yersinia pestis did not merely appear in history. Its lineages moved, shifted, and acquired traits that could make transmission more effective under the right conditions. The graves are not only evidence of death, they are evidence of microbial change. Picture the human side of that change. Roads connect towns, ports connect seas. Armies carry supplies and disease together. Caravans cross dry corridors with animals and grain. Warehouses store food that attracts rodents. Crowded neighborhoods place many bodies within reach of the same fleas, the same air, the same waste, the same fear. Civilization builds connection because connection is useful. It moves food, tools, money, news, marriage partners, soldiers, and ideas. The same connection can amplify a microbe. That is the deepest horror in the DNA. The bacterium did not need to hate its victims. It did not need intention. It only needed opportunities created by living systems that were becoming larger and more linked. A village contact zone creates one kind of opportunity. A Bronze Age trade corridor creates another. A 6th century imperial network creates another. A 14th century city like London creates another. The pathogen's genome records those changing stages in broken fragments pulled from human teeth. The first answer was simple. Yersinia pestis killed them. The deeper answer is colder. The graves preserve an evolutionary record of a killer learning to move through the world humans were building. That does not mean progress caused plague in some crude moral sense. It means density and movement have biological consequences. Grain storage changes animal behavior. Trade routes change exposure. War and hunger weaken communities. Migration creates contact between groups, animals, parasites, and microbes that may not have met in the same way before. Ancient DNA makes those consequences visible. A mass burial is no longer only an image of loss. It becomes a data point in the history of connection. A tooth is no longer only a personal remnant. It becomes a sealed archive of a pathogen's root, timing, and evolution. That is why the title's promise is so unsettling. Ancient DNA revealed that plague graves were not silent piles of bones. They were archives of migration, inequality, and social fragility. But the limits must stay clear. Not every mass grave is plague. Bodies can be buried together because of famine, warfare, execution, massacre, disaster, or ordinary cemetery pressure. A dramatic burial scene is not proof by itself. The tooth, the dating, the skeleton, the burial context, and the archaeology all have to agree before the claim becomes responsible. Not every collapse was caused by plague. Societies fail, transform, merge, migrate, and fracture for many reasons. Climate stress can damage crops. Violence can break communities. Political systems can overreach. Trade can shift. Hunger can weaken the young and old first. Disease may be one pressure inside a crowded field of pressures. The evidence points in a narrower direction. When Yersinia pestis DNA appears in a body from a particular time and place, it proves infection in that individual. When multiple bodies, regions, and time periods show related pathogen evidence, scientists can begin reconstructing broader movement. When that biological evidence matches archaeological signs of strain, the story becomes harder to ignore. Written records often counted bodies. They also blamed outsiders, described divine punishment, repeated rumors, or preserved the fears of educated survivors. Ancient DNA does not need those explanations. It identifies infected people even when their society misunderstood the disease, misnamed it, or turned its terror toward the wrong target. That is one reason the method has moral force. It can strip away some of the accusation and leave the biological fact.
A victim was infected, a pathogen was present, a route existed, a community was exposed. The rest must be built carefully, with archaeology, dating, climate evidence, burial practice, and human history placed beside the genome. The DNA is powerful, but it is not a complete witness by itself. It answers one set of questions and raises others. Still, the opening tooth now means something larger. At East Smithfield, the tooth named the bacterium inside London's emergency dead. In 6th century Bavaria, teeth showed that the first pandemic reached beyond the imperial story. In prehistoric Eurasia, teeth revealed that plague moved through human worlds long before famous chronicles gave it a name. A small object preserved the part of history that power forgot. The disturbing truth is not only that plague killed. People already knew that. The disturbing truth is that the victims most easily erased by writing can sometimes be recovered most directly through biology. The chronicles named the catastrophe. The teeth named the infection. The people inside plague graves were not a single mass. They were villagers, migrants, servants, children, laborers, elders, families, and strangers, buried under pressure when normal ritual could not keep up. Some died in cities, some died near routes of movement, some died in worlds that left no written witness at all. Ancient DNA does not make the past cleaner. It makes it harder to ignore. It tells us that behind every plague number was a body with blood moving through it. Behind every emergency trench was a community trying to manage fear. Behind every famous pandemic was a wider field of unnamed people whose bones and teeth carried the evidence when paper did not. That is the work of the Genome Archive. Careful genetic history, ancient DNA mysteries, and the human record preserved in bones, teeth, and blood. Subscribe for the stories written below the level of kings and empires, where the evidence waits in the parts of the body history almost missed. No hierarchy, no curse, no simple collapse story. Only evidence, movement, survival, and memory.